What are we going to cover today? First and foremost, we're going to make sure that we're keeping the sample healthy because there's no point in doing live cell imaging if you kill your sample with just the imaging. Next, we're going to go into what are some of the hardware options that you want to look to that will make you most successful doing your experiments. We're going to go into a few techniques for labeling your sample. Next, we're going to go over the different systems that are set up for live cell imaging in our facility and what are some of the unique features that they have that will make you want to do specifically that one. And then we'll have a moment to touch on advanced experiments, but you don't want to be here for forever, so I can't get too much into that. So let's first off with keeping your cells happy. There's lots of names for this. I like to call it pyramid of pain. And along with normal imaging, where you need to have your spatial resolution, signal to noise, and speed, and you can generally only pick 2 of those, you also need to, first and foremost, keep your sample healthy. So this is going to involve environmental, so heating or CO2, or we have people that do zebrafish, and the system might still be warm for another experiment. Zebrafish don't do well at 37 degrees Celsius. Phototoxicity, so this is keeping our cells healthy, and then what we want to do media-wise, because media can actually affect what your image quality is. So first thing, a lot of our systems that are designed for live cell imaging have these massive boxes around the systems. And these work by, we have a heating unit that's forcing warm air into that entire box. And then on the other end, we have a CO2 tank that's being mixed to be your 5% if you're working with mammalian cells. It comes up, bubbles through a humidity bottle so that we are having generally over 90% humidity right where your sample will be. Why is this humidity important? Well, if we don't have the humidity, we're going to have evaporation of the media, which is going to change the salt levels in your media, as well as the pH. The other thing with this is with the evaporation, my tip for it is when you're using a multi-well plate, if you don't need all the wells, leave the outside wells open. Those are going to evaporate most quickly from your samples. Actually add water to that. Then that water on the very outside will evaporate and your sample will stay happy on the inside. You also, if you've tried this live cell imaging with us, we'll tell you, turn on the incubator before you want to image, let it warm up. And the reason why is with these large incubators, this is a graph specifically, Pecan, our black incubators, their graph, that the first 30 minutes, you're having a very rapid change from room temperature up to 37 degrees Celsius. Why does this matter for your imaging? While this ramping is occurring, you're going to have thermal drift. And so you will start in focus and it will slowly move out of focus, just like a bridge expands and contracts in different temperatures. Even though this is going to be a small movement, where we're staying in focus is a very small amount. So next, once our sample is like it's being in an incubator, if that's what you need, phototoxicity. So you've probably heard this thrown around, but you may not know, what does this actually mean? So this is going to be damage to your cellular macromolecule with your excitation light. How does this happen? Well, it depends on the wavelength. If we're using 405 light or UV light, this can directly cause an apoptotic response or DNA damage directly. If we're using the longer wavelength excitation, we still can produce reactive oxygen species either by the fluorophore or endogenous molecules. This results in ROS, which is going to oxidate your proteins and affect your cell health. So what are some of the things we can do because we don't want phototoxicity? Well, one of them is actually in your sample hardware design right here. If possible, you want to use the longer wavelength fluorophores. This is because this is less energy that is actually coming to your sample, so it is most gentle on your sample. So think using your reds are going to be best. We also want to lower photon dose. This can be done in generally 3 ways. You can use less weight laser power. This is the obvious one. Depending on if you're on a wide field or a scanning confocal, shorter exposure or scan faster. This is going to have less photon dose per area, or fewer frames, fewer Z planes. What does this mean? Well, when you go into our software, there's going to be optimal Z-stack, and that's based on physics and Nyquist sampling. But in reality, if you're trying to keep your sample healthy, all you actually need is to sample your feature of interest twice. So if you just need to know you're in an organoid and you need to be able to count all your nuclei is probably going to be around 5 micrometers. A 2 micrometer step is really all you need. You don't need those some 1 micron steps. Other things we can do is if we don't need to take images every 5 seconds, if you can get away with every 30 seconds, this gives the chance for the cell to actually antioxidize, I guess would be the word we're going with, and let those come down in between. And finally, lower concentration of probes. This depends on if you're a dye, you're going to do a serial dilution to figure out what is the lowest level you can use and still get good results. Or if you have fluorescent proteins, you may have different expression levels in different cells. You don't want to pick the cell that's blazing. That cell's going to die real quick. You want to pick the mid to lower expressing cells. Okay, so media. There's going to be 3 things we talk about with media. First and foremost is cell health. So this is going to be your monolayer cells, organoids, is where this comes into play. So in this graph, the very top row is just your standard complete media. You can see as it's imaging over time, the cells are growing, they seem to be happy. I'm going to turn down the light a little bit so we can see better. And then in between these last 2 panels, they're withdrawing the CO2. You can see all the cells pretty much die off when you do that in complete media. If you do serum-free media, they pretty much are not replicating and they die off real quick. And then this is the final one we're going to focus on. If you have complete media plus heaps or heapies, depending on how you pronounce it, your cells are going to grow over time. You can remove this CO2 and your cells are going to continue to be happy. What does that mean? Some of our systems we can put in a heated stage, but we don't have CO2. And that means that you can do some less long-term imaging, but you can do imaging without the CO2 for some time. So then you can expand which systems you can actually image on. For our zebrafish folks, you don't need all these things, but it's good to know. Okay, media, photosensitizers. So these cells are transfected with GFP. Our top row is the very first image that is being taken with these. And just straight DMEM right here. We start off with not much in the red channel. After 20 seconds of continuous imaging, we have photo-converted a lot of that into having a red emission. So if you didn't run your controls and you did this, you might think, I have co-localization. But in fact, it's actually just photo conversion. A cheap thing that you can do is rutin. It is an antioxidant, relatively cheap to buy. You do have a little bit of autofluorescence in the red channel, but your cells are going to stay happy throughout that imaging. They do make specific medias that will prevent this photosynthesis. So it's not a photosensitizer. That you can see it stays green. We don't have anything in the red channel. Okay, last thing on media. Media can greatly affect autofluorescence. So in the first graph, we have fetal bovine serum. Most of the people working with mammalian cells will have that in their medium. This curve right here is for UV emission. You probably wouldn't be doing that for live cells, so you don't worry about that as much. But this light blue right here, this would be what you're doing in your GFP likely. And you can see this autofluorescent curve that we have with the fetal bovine serum. Next, we have a spectrum with phenol red. And we again have this curve that's pretty significant in the red and the green and the red emission. So what can we do about this? There are different medias, such as Optimum, that are actually designed that you can use 50% less serum for the duration of the imaging. And your cells will stay happy. And the very least that you can do is get whatever medium that you're normally using without the phenol red, and this will greatly improve. Why do we not want autofluorescence? Well, your best imaging is going to be when you have a high signal and low background, and this is bringing up your background so that you're going to see your signal less. Okay, next on to hardware options. So what are we going to do about focusing? Which objective should I use, and how am I going to mount this? So many of the systems we have, if it's Zeiss, it's going to be definite focus, and then our Nikon spinning disc has the perfect focus system. This works by shining a far red LED. It's going to hit the surface of your sample. It's coming back down, and then it's measuring what that distance actually is. So this is going to help if you're having any thermal drift over time. But it's not going to help you if your sample actually moves away from the cover slip interface. There's not a whole lot we can do if your sample moves on us. So things like zebrafish, we suggest that you mount in a low melting point agarose, so it doesn't actually move on you while you're imaging. Okay, objective choice. This is a paper from Aaron Deal, who was 2 before Alex, I want to say. And this is looking at refractive index mismatch. In this case, it's looking at if you were using a water immersion objective with a sample in a hard set mounting media. But the reverse is true as well. So if your sample is this purple, and it should start here and there, if you have a refractive index mismatch by using an oil objective and an aqueous sample, what you actually see is going to be different. You're either going to have a compression or expansion, depending on what mismatch you're actually doing. And this means if we are doing 3D imaging for live cells, we want to be using a water objective for having your best Z resolution. You can get away with using an oil objective if you're looking at something that's just the interface between the cover slip and the membrane, then it doesn't matter as much. The next thing with objective choice is your working distance. People forget this all the time and then ask, why can't I get my sample in focus? Well, working distance is inverse to the numerical aperture. And most systems, as the magnification goes higher, we're also going to have a higher numerical aperture. We have several objectives that are specially made to be long working distance objectives to counteract this. But how does this become a problem? This is my terrible animation. So if you have a water objective, this is your working distance. Right now it's focusing on the bottom of the spheroid. As you can, I made it so that the objective would hit to the bottom of the plate. We still have a little bit of the spheroid we're not getting through. So we need to look at this beforehand. And I have made a spreadsheet that we all share with the different working distance of our objectives. So if you have something really big, like a fish or an expanded sample, come to us and we can tell you, are you going to be able to make it all the way through your sample or not? Okay, mounting. So which system you can use can be dependent on what you can do for mounting your sample. For any of our laser scanning confocals, such as we have 4 900s and a 980, you're going to have an inverted position in which we have a dish or a slide that has this number 1.5 cover glass bottom. If you come in with a tissue culture plate that has a millimeter of polystyrene, the best objective we can use is a 10x, and it's still going to be blurry because that polystyrene is going to bounce that light everywhere. We also, the Lyra and the Nikon spinning disc will also be using this method. We have plate holders as well, so it doesn't have to be dish or cover or chambered slide. We can also do multi-chambered plates. Okay, for our fish or organoids people, this is where you might go to the light sheets. We have 2 different light sheets. There's the Zeiss Light Sheet 7, Bruker TrueLive. This would be in an agarose column, and we image from the side. Or if we're on the Bruker light sheet, we have this trough that you just place your sample in and image from the bottom. 2 photon, this is most of our organisms end up on this system. And we have this water dipping objective, that the objective is actually going to go right into your water-earned media. What you need to take into account is that the barrel of this is quite big. We suggest 50 millimeter or bigger plates. It does not need to be a glass cover slip bottom, because you can see we're imaging from the top. Whatever the bottom is does not matter. On the 2 photon, we also have this water immersion that you can have a glass cover slip on if you want. This is only a dot of water as opposed to actually dipping it into the sample. So that's another option for it. But obviously you need to have the number 1.5 cover slip again. Okay, moving on to your actual sample. Labeling your sample, you're going to have a large variety. Wanted to point out, because not a whole lot of people use it, but it can be quite useful. There is, we can do transmitted light on our spinning disc, as well as both 900s have cameras on it that we can just do wide field transmitted light. You can see a lot with this. But if you need to label your specific proteins, we have 3 main options: your fluorescent proteins, dyes, and tags. So what is that? What do you need to know about these? Fluorescent proteins are your genetically encoded option. They can either be stable or transient, depending on if the plasmid integrates or not. You can do a wide number of targets. There's a large palette. You can just order some off Adgene. You don't have to make them yourself these days. But the problem with this is if you're doing an organism, it could take a long time to make it, or we have some people that are actually doing organisms that are not genetically tractable right now. What do you need to know about the fluorescent proteins? So this right here is from fpbase.org. It's a really useful website for you to look at your different fluorescent proteins. Right here we have our curves. This is our excitation and emission curve. On the top, it's telling you what are all the family members before it. And you're going to have your excitation and emission. And then this EC right there, this is your extinction coefficient. And this is how strongly a molecule will absorb light at a given wavelength. And this is being measured right at its maximum excitation. Your quantum yield is how likely that molecule will then release that photon after being excited. So then to be able to easily compare it, we have this brightness number right there. In this case, it's 78, which is Scarlet 3 is one of my favorite. It's super bright. And then the other thing to really take note is this maturation. This is how long it takes for it to fold and be fully fluorescent. In this case, it's 37 minutes. The longest I've seen is 24 hours. So there it can be, and then I've also seen as low as 12 minutes. So when you're making something, if you're actually looking at putting a promoter of interest in front of your fluorescent protein and seeing when that gene is active, if you need to measure 30 minutes after, but it's not holding for 4 hours, you're not going to get an accurate representation of when those promoters are on. So there's a whole lot of cell permeable dyes to measure pretty much anything. I found this Thermofish or Stain It's pretty fun and useful, that you can tell it what structure do you want to label. Do you want, what type of product do you want? What emission are you putting on it? And then it will give you a drop down of several different products that would fall under that category. In this case, I used all the possible excitation emissions that you could feasibly put together on our LSM 980. Tags are somewhat related to our dyes, but work a little bit differently. Most commonly used ones in our facility are our snap tag, clip tag, and halo tag. A lot of them are very similar. And one of the big things to look at is snap tag and clip tags are both 182 amino acids, while a halo tag is 297. So if the size matters for this, then you may want to pick a different one. The dye that you put in can be agnostic to which one of these you use. What I really suggest to have your best imaging is to pick a dye that is non-fluorescent until the reaction has occurred. Otherwise, you may have a lot of background. So some of the ways this can happen is that you just have a fluorophore that until the snap reaction occurs and kicks off some of the molecule, there isn't fluorescence. Another is there's what's called a quencher. So the fluorescence can't occur with that nearby, and then it is removed and fluorescent. Finally, it's not fluorescent in, say, a hydrophilic area, but once it ends up in a hydrophobic binding pocket, it will now be fluorescent. Yep. Doug, correct me if I'm wrong. I've seen people do two together.
Yeah, so it's kind of like a hybrid between a dye and a fluorescent protein, right? You still have this big fluorescent protein tag that you have to have there. So you could put a snap tag on one protein, a halo tag on another, and direct different dyes to those.
So the answer is yes, you can combine them together. Okay, so now we're going to go over which samples should go where. Generally, we're going to separate samples in our facility into 4 different categories. Are they thin? Are they thick? And are they moving or not? So right now, we're going to just go over thick and thin samples that are dynamic. So in general, big samples are going to go on our light sheet or 2-photon. The 2-photon has a lot of working distance and large field of view to help accommodate our big samples. And then our small samples, we have our LSM 900 live cell has incubation. We are going to refresh the normal LSM 900 shortly, and that will also have an incubator. We have an LSM 900 with incubation over in Alston, if you don't remember that we have this facility in Alston. The LSM 900 and Lyra both have incubation as well as the spinning disc. So let's first go over why you'd use the Bruker TrueLive. Well, what do you need to know about it first? Hardware. We have 405, 488, 561, 647. So this is your DAPI, GFP, RFP, far array. There's only a single objective. So you don't need to email us ahead of time to tell us which objective that you want to use if anyone's used the SICE light sheet. But there is an optibar. So you can change what the actual magnification in field of view is that go from 12.5 up to 50x. This has full incubation, dual camera. Dual camera means you can do 2 channels simultaneously very rapidly. With these cameras, you can do ultra fast imaging, 40 frames per second. Full chip, no binning. At this rate, usually what is the defining characteristic is how bright your sample is. Can we get enough photons in that very short exposure? We've had people do up to 72-hour experiments with organoids in here. So you can keep your sample in here for a very long time. And with this trough right here, that's just an aqueous media of whatever you want, no embedding is needed. So for organoids that if you wanted to do on the Zeiss light sheet, if you had to put it in the agarose, there's the possibility that your cells are going to grow out of that organoid into that agarose. This, you're just dropping it in so you don't have that problem. Or if you have an organism that does not want to be embedded in the agarose and you want to keep it alive, this is what you do. The only thing is be mindful that the sample can't get too high up in the trough or imaging from below. Zeiss light sheet 7, we have the same ability of fluorophores to be done. We have a variety of objectives. For our water objectives, if you're doing live cell, we have a 10x and a 20x objective. And then this can be done with water samples. We have a 5x objective, so this is going to be your much larger samples you're going to do this with. And then just to note, we have a 20x objective that can be for clearing solutions, but clearing and live cell do not go together. It has full incubation and dual camera again. You're going to see kind of a theme that we have live and we want to go quickly and we're going to have more than one detector. So what is special about this is you have multi-view experiments. What does this mean? Well, under normal circumstances, you're going to illuminate and then you're only imaging from one side, even if you're doing dual illumination. So that front side right at the objective is going to look really nice. But as you go through, it's going to get blurry. In this situation, we can actually rotate the sample with the machine and image from multiple views and give you that same great resolution throughout your sample. So developing embryos were great for doing things like this. Okay, next, spinning disk for our Nikon. So we have our common 4, but we also have where this is an orangey red, this is more of a red. So we have an extra laser line on that. Objectives, we have 4, 10, 20, 25, and 60x. So the 4x would work really great if you have an organoid that you want to be able to get through, and you don't need a really high resolution. And then there's 25x 1.5 silicone, has less working distance, but it would be really great for any sample that's close to the objective. It's going to be what's closest to a water refractive index on this. It has full incubation, dual camera, and also has photo manipulation at 405 and 473. So if you want to be doing optogenetics or anything like that, that's possible on this one. We're again having ultra fast imaging. This camera is slightly faster with 43 frames per second. And if we crop and bin this and you somehow have enough light, so imagine cilia meeting in bright field, you can actually do 500 frames per second on this system. Just be prepared for the data size on the other end if you're running that. This is the only system in the entire facility that you can do hypoxia experiments. The Fishman lab bought this bought the incubation setup for this. So you can actually change your oxygen levels with that. It does normal CO2 and oxygen as well. And then I just wanted to point out, this is down in this is over in Sherman Fairchild in our live cell imaging facility there. Okay, we have lots and lots of LSMs in our facility. They're well loved. So this is going to compare our 980 versus 900 live cell. A lot of people, they love the 980, but they can never get time. Maybe you don't need it. So let's go over what are the differences. If it's not underlined, it's the same throughout. So we have the 405, 48, 561, 640 that we have almost on every single system. This one, we also have stuff for CFP, YFP, Ready Red, as well as size 7. And we have the detectors to do that. Objectives, We have the same 10x objective. We have identical 20x objective. We have identical 40x water objective. But on the 980, we have a different oil objective, and there's a 63x oil. On the 900, we have a low magnification 5x. So if you need that longer working distance, that's going to be great. And just a different 40x oil on that. Okay, differences in detectors. Both have TPMTs, both have 2 PMTs on there. Those are identical. Our gas detector on them are slightly different. Both have a gas detector, but the Zeiss has a 32-channel gas array. And then it has an area scan, and then it has the near IR detector. So what does this mean? Both have full incubation, both have full incubation, both have definite focus, both have similar objectives and detectors. Yay! But what unique applications can you do that you cannot do on the 900? So area scan and area scan fast is going to be just your 980. Spectral detection, so that's your 32 detector array. We can have multiple fluorophores that would normally be bleeding into each other. We can acquire those separately and unmix those. And then there's Dynamics Profiler that Linda will be presenting on July 11th if you want to look in any real dynamic processes. Okay, Zeiss Lyra, there's several different modes that you can run on this. Laser wide field, you're going to use if you need something really fast. Camera-based system, so it's not going to be as slow as a laser scanning confocal. TIRF, you would use for very low photon dose. And then you can see right here, this would be your wide field versus your TIRF. This is really going to bring the signal down and give you really good imaging if you're right at the surface between the cover slip and the membrane. And then we have this SIM mode, structured illumination microscopy. This is giving you approximately 2 times the confocal resolution. Want to point out though, when we're doing these super resolution microscopy, these numbers here is the fold increase compared to a standard confocal. If we're doing SIM in 2D, we're having 4 times the photon dose. In 3D, we're now doing 8 times the photon dose. So we have to be really careful with our phototoxicity if we're trying to do SIM and wide field. Our STED system, technically you can do live cell, but you can see that we're now having 100-fold dose. Okay, so what are some of the more advanced things that you may do here? So these are generally going to be different dynamic processes. We can do FRAP and FRET on every single confocal, for fluorescent lifetime on the 880, fluorescent correlative spectroscopy on the 880, and then the dynamic profiler is on the 980, and then the 900 bio over in Alston. So what are FRAP and FRET? Let's go over those because those are the most common things that you'd be doing. So this stands for fluorescence recovery after photobleaching. You're going to use it to determine the kinetics of diffusion. This is generally going to be used for cell membrane kinetics and protein binding dynamics. So what is actually happening? This is time 0. Then you intentionally bleach out that uplink on your sample. And then look at how long does it take for you to regain fluorescence. And these individual molecules generally aren't regaining fluorescence if this is from diffusion back into that area. And so this is the curve right here. This is your intensity. You're dropping off when you're bleaching and then following that circle again until you have recovered. Generally, you're never going to have 100% recovery. Next, we have a lot of people that want to do FRET, which is Forrestor, or a lot of people will call it fluorescent resonance energy transfer. This is generally going to be looking at protein interactions. It has to be very close protein interactions. Or you can use linked fluorescent proteins that are used as biosensors. So what do you need to have sufficient FRET? First and foremost, you need to make sure that where the donor, this is going to be your lower wavelength fluorophore, emits in an area that will accept that will excite the acceptor. This is your longer wavelength fluorophore. If you don't have this overlap, you're not having FRET. Next, it needs to be within 10 nanometers of each other. Any more than that, you're not getting this transfer of energy between the fluorescent proteins. And then finally, you need to have your correct orientation. If the orientation is wrong, you're not getting this FRET again. So this is why it can be quite difficult creating a FRET sensor all by yourself. We suggest trying something else first or buying one that someone else has already made and tested and proved it works. What are some fluorescent biosensors? Almost anything can be sensed these days. So ion concentrations, oxidative stress, pH, cell cycle, and a variety of other things. If you're interested in this Specht et al. 2017 really suggests you checking that out. They go over a large number of them. So what is most done in our facility is calcium indicators. And I like to put them into 4 categories. See, I like categorizing things. So you have your genetically encoded or dye-based, and those are either going to be intensity-based or ratiometric. So intensity means when the calcium is present, that intensity goes up. Ratiometric is generally going to be, you're going to have a shift in spectra when this actually binding occurs. So most commonly genetically encoded one is this GCAM. Ratiometric, we have this chameleon that you have a cyan fluorescent protein and a yellow fluorescent protein. So this will take into account for any difference in expression that you might actually have in this reporter. Flow 4 can be used, and it's again intensity based, and you just have to make sure that the difference in intensity that you see is actually calcium related. I had a situation for a ROS sensor when I was at Wake Forest that we thought there was more ROS in some areas, but it turned out that it was actually differences in permeability of the cell wall between these different cell species. So try to avoid our artifacts as much as possible. Indo-1, it's the only ratiometric dye I know of. Unfortunately, it's excited at 338. The lowest wavelength that we have is a 405. Also, that UV is not really that great for your sample. So we won't be able to do that if you come in with that. Okay, so every microscope has its strengths and weaknesses. And to get there, you need to determine what is the absolute minimum resolution you can have. When you're doing live cell imaging, you might not be able to get that beautiful image you want. What is the speed of the biological process? Cells dividing versus calcium firing are way different time frames. How long do you have to image to be able to see your phenomenon? And then what is the physical side of your sample? These are all the things you need to ask yourself before finally deciding which microscope you want to use. This is from St. George's University of London, so we don't have exactly the same systems as they, but they're categorizing things by speed, depth, resolution. Wide field's going to be really fast, but you're not going to have good depth or resolution. SPIM is another name for light sheet, where it's again fast, we're having better resolution and depth. Laser scanning confocal is kind of mediocre on every point. Multiphoton is going to be really good for getting in deeper into your sample. Our super resolution is going to be slow, but good resolution. We don't have a spinning disk super resolution, we just have a spinning disk, so less of that resolution, but fast and decent depth into your sample. Okay, I'm finishing up, I promise. Some resources. fpbase.org is really great for looking at your fluorescent proteins. Your molecular probes handbook will have all the different dyes that you really need. Adgene has a blog, and I've actually found that Doug has a blog post called "Which Fluorescent Microscopy Technique is Best for Me?" And you get to see a picture of young Doug if you go there. Come talk to us. I would love to talk to you about your live cell imaging if you couldn't tell from the talk. Image.sc is where I would suggest to go if you need any help with the analysis on the other end. And then Bioimaging North America, under their training and education tab, have put together a whole lot of different video resources as well as workbooks and web pages that could be useful. And, yeah. And since you've put up with me this long, you guys get to be the first ones to hear. August timeframe is what we're hoping for right now. A Yokogawa CV8000 will be coming. So this is another spinning disc, designed mostly for plates, but we can get most samples in there. It's going to be able, it's a high content analyzer, so it's going to automatically go through, take images of your sample, like the Cell Discover, except it has a spinning disc, so you get that combo resolution that you won't be able to get with the Cell Discover. And hang out here. What questions does everyone have?